This is for Biology 223 Genetics. We have an article published in Cell in 2016. It's a bit wild, and I want to compare it to an article in Cephalopods published in the year later. That's interesting. So this is forefront of genetics. Doesn't relate to a specific chapter, perhaps chapter 7. Long neural genes harbor recurrent DNA break clusters in neural stem-progenitor cells. So it's a complete sentence here, and it's a statement. Their finding is that they found that these specific genes harbor DNA break clusters. Okay, so its significance may not be immediately obvious. Let's look at the summary, the abstract here. Repair of DNA double-stranded breaks by non-homologous end joining is critical for neural development, and brain cells frequently contain somatic genomic variations that might involve double-strand intermediates. So that's a pretty serious sentence here. So we have double-strand break repair. Double-strand break repair is necessary just in the normal course of mitosis and meiosis. It occurs when DNA damage is detected, and it's something where the DNA, if it is broken spontaneously or by whatever cellular mechanism, it has to be reconnected. However, if it's done incorrectly, then it can lead to rearrangements that's described here. So generate chromosomal rearrangements, translocations, deletions, inversions, amplifications, which can be very dangerous and carcinogenic. So maybe this relates to the genetics of cancer, if anything. Okay, so they are telling us that these double-strand breaks by non-homologous end joining, it's critical for neurodevelopment. That's a little bit strange. We would understand by repair of double-strand breaks is important, by non-homologous end joining might be important for survival of cells when it can't be done by homologous, but that it's critical specific for neurocells might seem a little bit unusual. And brain cells, as opposed to other cells, frequently contain somatic genomic variations that might result from this way. So they're alluding to a hypothesis that neural function, that is brain intelligence, requires a genomic mosaicism. That's a really bit of a wild thought. We're not surprised that brain cells might require a different sort of repertoire of expression and regulated by special mechanisms not found in other tissues, but that the brain has a genomic mosaicism, that's quite interesting. It implies that intelligence, behavior, sophistication requires a whole nother layer of not just regulation, but a variation that's not found, that our genome in and of itself, and that's reminiscent of immune cells. We see also here that they will say that this non-homologous end joining double-strand break repair is required for both immune and nervous system development. Okay, we know that. That's in the book in development. VDJ recombination for antibodies. There's somatic mutation and there's rearrangements necessary for immune system diversity. Okay, so we understand why some sort of change to the DNA in somatic tissues is necessary for the immune system. Why it's necessary for a nervous system is a bit more surprising. We now use an unbiased high-throughput approach, so it's a new technology that developed here, to identify genomic regions harboring recurrent double-strand breaks in primary neural stem/progenitor cells. So in these neural stem cells, they're specifically looking at them, which are going to develop into the brain. We identified 27 recurrent double-strand break clusters. We refer to those as clusters. And remarkably, all occur within gene bodies. Okay, they say remarkably. Why remarkably? That they find if these were randomly distributed, then we wouldn't expect them gene bodies. Gene bodies make up a few percent of the genome, so if all 27 occur within gene bodies, it implies that there's some mechanism having to do with the gene body that they're being transcribed, or there's some other mechanism that identifies specific genes that are necessary, that are going to be subject to this non-homologous end joining, that are important in brain function, and that's the source of rearrangements. That's a little bit wild also. Most of these clusters, recurring clusters, were detected only upon mild alpha-dycoulin-induced replication stress. That's a drug that interferes with helicase or resolution, so it's a stress that causes double-strand breaks to occur, and they did that because the frequency is so low that they wouldn't see it. So this is something that's very rare, and they use it's topoisomerase, I forget which one. This is an inhibitor just to increase the rate so it can be detected, and presumably it's unbiased, providing a nucleotide resolution view of replication-associated genomic fragile sites. So this is where does the genome, is it likely to break? We have a long genome, and when we look at double-strand breaks and where rearrangements occur, it's not randomly distributed. There are fragile sites. What makes a site fragile? That's the sort of thinking we should be doing. The vast majority of recurrent double-strand break clusters occur in long transcribed, so long, that is, they're large genes. Even if the open reading frame is relatively short, if the processed mRNA is relatively short, the actual gene body, the part that's transcribed, could be quite long. It could be hundreds of thousands of base pairs long. So long genes, transcribed genes, that implies that the gene must be transcribed, or it's transcribed in these particular cells. And the idea is then that the mechanism by which these breaks are generated could involve transcription, in which we do have a need for topoisomerase and gyrases and helicases, because the transcriptional machinery proceeding, and so that's a source of stress. And late replicating, when do they mean? Late replicating in S phase. So in S phase, of course, and there's a problem earlier in the book. When is that? Maybe chapter 4, 3, 5, about how we identify S phase and how we identify when a gene is replicated in S phase. And so late replication, you can imagine there's more stress involved late in S phase. Anyway, that idea is a bit more complicated. Moreover, almost 90% of identified recurrent double-strand break cluster containing genes are involved in synapse function and or neural cell adhesion. So these genes are active in a specific tissue in the body. They are long, and they are actively transcribed, and they're late replicating. So that means it looks like this is a specific mechanism that is evolved for a specific purpose or selective advantage. So synapse function and neural cell adhesion, which is the mechanism by which our behavioral sophistication occurs, with a substantial fraction also implicated in tumor suppression and or mental disorders. What does that mean? Well, that means that gene is implicated in mental disorder. It means its absence causes mental disorder, which implies that if these genes don't undergo rearrangement, if this mechanism of genomic mosaicism doesn't occur, it can cause a mental disorder, that our intelligence and normal mental functioning requires this genomic mosaicism. Our characterization of these neural stem cell progenitors, recurrent double-strand break clusters, reveals a basis of gene fragility, so the mechanism by which the breaks occur, and suggests potential impacts of DNA breaks on neurodevelopment and neural functions. So that's not really strongly stated. It's alluded to. They're saying what they want to say, but they're not emphasizing the rather remarkable ideas here that our intelligence requires relies upon a genomic mosaicism that's generated by something that's very dangerous and likely to cause tumor genesis as well. So the paper is very well written. It's quite long. It's well written. It has a nice introduction. Some of it's technical, but it's accessible, and it's a very nice example for us to see what the forefront of genetics is. I will end here.